Hello, welcome to our meeting for this week. Because of some technical issues, I'm doing the update by myself, so welcome. I'm going to cover some tools that I learned about at the OpenWorm annual meeting. I'm also going to go over several papers that we have in our queue. So let's get started. So this last week was the OpenWorm annual meeting. The DevoWorm presentation went successfully. We got a lot of good feedback. People were positive about our work, which is good. This is a meeting of the Board of Trustees of the organization, plus all of our senior contributors. So we have some interest in our work. Now, there was another group. There are two groups that are really doing a lot of work in OpenWorm right now. Our group and Porrig Gleason's group. Porrig Gleason is based, I think, at the University College London. And he's been doing a lot of work with the Connectome and getting together a lot of secondary data. So he's involved with INCF and their different initiatives for data sharing. And then he's also been working with someone on building a large language model for the C. elegans literature. So the first tool here is the C. elegans Connectome Toolbox. This is from Porrig's group. They've been working on bringing together all of the major data sets for the C. elegans Connectome. We have the gap junction data sets. We have the synaptic data sets. We have the neuropeptidergic data sets. We also have the hermaphrodite data set and the male-specific data set. So this is the tool here, OpenWorm.org Connectome Toolbox. You have an overview, which is here. This is just talking about sort of example plots. So when you have a Connectome data set, how do you plot it? I think that's an interesting problem in light of some of the discussions we've talked about plotting network data out. So, at the top we have sort of our hairball slash it's not really a force-directed graph, but it's in some sort of hairball slash force-directed context. You have hive plots on the lower right. You have a circuit diagram in the lower left. And you have sort of this heat map at the upper right. So it just shows different ways to plot these interactions, these ith interactions. And so that's, and I think it's an important thing to think about, especially with connectomes, because you're trying to communicate two things. You're trying to communicate the interactions between neurons, and you're trying to communicate the function, perhaps. You're also trying to communicate something about the structure. And so if you have different modules, different circuits, if they interact with crosstalk, if they're separate or inseparable, then those are all good things to know from the data. Some plots are easy to interpret and some are not. So this is an ongoing area of interest. Then we have these historical publications. So this is a nice chart. This actually goes into sort of the different categories of Connectome and data sets, and then the publications associated with them. So we have the anatomical, chemical, electrical networks. We have the extrasynaptic networks. We have the gene protein expression networks, and we have the functional networks on the right. So we start with Albertson and Thomas in 1976. We move to White 1986, Durbin 1987, Hall and Russell 1991. That's all in the anatomical, chemical, electrical category. So this is where they're mapping out the basic features of the connectome, the cells, the connections, etc. Then we have, in gene protein expression, we have Altoon et al. 2009. Goes down to Taylor, Yamini, and Beets in 2021 and 2023. So that's actually quite recent work. And a lot of these other works here on improving the connectome are also from 2010 on. So Varshni 2011, Cook 2019. This is the gap junction connectome of the hermaphrodite in the Connectome of the male, respectively. Then you have Cook 2020, Britain 2021, and Whitfleet 2021. We have additional extrasynaptic networks, or actually, we start with our extrasynaptic networks in 2013. Serrano Says, Pereira, Tendril Atlas, and Hobart, Bentley, and Rapoli Sanchez. So this is actually where we're talking about the peptidergic networks. Then in functional networks, we have these 4 papers. So, they're all from 2020 on. And so they have some nice visualizations of the literature, of the data, its provenance, what it is, and so forth. So I leave you to explore that on your own. We have access actually to the data sets here. We actually have, I think, you can get to the actual data set here. Okay, so this is like a stub for the data set. We actually have XLS files or Excel files. I think they're also data readers and things like that are available from different sources. Yeah, so a lot of these are stored in Excel files, and there's a reader here as well. So if you want to play with some of these connectomes, they're all collected here in the Connectome Toolbox. Okay, so that's the Connectome Toolbox. Now we have what they're calling OpenWorm.ai, and so the idea here is to take a large language model, and this runs actually on Hugging Face. The URL is OpenWorm.ai, so you can reach it through there, but it's actually run off of Hugging Face, and it's hosted there with a number of large language models that you can use that are trained for this purpose, which is ask individual large language models questions about C. elegans. You ask them about something from the C. elegans literature, and you get an answer coming out. So this is the individual LLM stub. Like if I ask, for example, what is the primary role of the C. elegans neuron AVBL? And I want to use Gemini, and the temperature, which is a parameter, is 0.1. And you can't really uncheck this box, but only answer questions related to C. elegans, because sometimes you can get things that are not related to C. elegans. So if we submit this, we get an answer. The primary role of the C. elegans neuron AVBL is to mediate thermotaxis behavior, which is the movement of the worm towards or away from a thermal stimulus. So it gives you the function of AVBL. I can also choose other large language models, Llama 2. Let's try Llama 2 just to see what happens. Okay, now that's a little bit different question or answer. A great question. The primary role of the C. elegans neuron AVBL, known as AVE left, is to function as a sensory neuron involved in identifying and responding to a broad chemosensory cues, particularly food sources such as bacteria and their associated volatile compounds, VWFA. Specifically, AVBL neurons are responsible for detecting these chemical signals through specific receptors on their surface and transmitting this information to other neurons in the neural circuitry of C. elegans to guide locomotory, and then it stops. So, you can see where you can find out a lot of different types of information, a lot of things in parallel, and then that's kind of your answer. Okay, this is a little bit different, but it basically gives you the same answer. So this is, playing around with large language models, asking questions about C. elegans biology, and I've tried it with other questions and it works adequately. There's this panel discussion, so this is where I guess you can actually have a panel of large language models. So let's pick I guess you can only do one to lead the panel. So let's have Llama 2 lead the panel. And then Gemini, GPT-4o, GPT-4, and Cohere. Let's have them. So it's like you have this leader of the panel and then the attendees. And I guess they're all going to give some sort of consensus answer. So let's submit that. We'll see what happens. It might take a while to do. Okay, so it gives me a number of answers. So Llama 2 will say, based on our expert panelist responses, it appears they are in consensus that the typical length of C. elegans worms is approximately 1 millimeter. I don't know how that relates to our question here. The typical length of the worm C. elegans. So this is the question they're trying to answer. However, I notice a slight inconsistency in Dr. Gemini's and GPT-4's responses, which do not explicitly state they're referring to adult worms. So there's a lot of sort of semantics and semantic guessing here, which is typical of a large language model. So yeah, Gemini says 1 millimeter. The typical length of the adult C. elegans worm is about 1 millimeter. GPT-4o, the typical length of the nematode worm C. elegans is about 1 millimeter. And so it's just kind of giving answers that are kind of more or less specific to, say, like an adult, or some variations in the name. So it's just interesting. I think if I asked it a more complex question, it might be able to give me a more interesting answer. Okay, so now you can actually search for publications. C. elegans locomotion. If I want to get 10 results on C. elegans locomotion, then I have these search results. It basically goes through the literature it's trained on and pulls out literature. And it gives like a sort of an annotated uh, literature review where it says, it gives some description of the article and how relevant it is to you. Structured data, so you can actually query different types of sources. This is the Worm Neuro Atlas. Which neuron would you like to know more about? AVBL. So if we look at the answer here, we see that it gives information about neuron AVBL from Worm Neuro Atlas, and it looks like it's formatted data, just giving information about the anatomical connectome, its inputs, its putative inputs in the neuropeptidergic connectome and then the anatomical connectome, and then it's giving outputs as well. So this is really interesting. I hadn't realized that there was this kind of structured data aspect to it. The OpenWorm corpus, of course, it will give you a list of specific cells for, say, like the pharynx. So if I say, what are the main types of neurons in the C. elegans pharynx, it gives me these neurons. And, it cites it's where it's growing the data from the Worm Atlas handbook. So this benefits from the vast number of community resources we have for C. elegans in the community. We have all sorts of resources, shared data sets, and other things that we can actually put into the large language model to query. So this isn't coming from nowhere. This isn't just coming from the literature. The community has a lot of tools that they can use. The problem, of course, is that they're all kind of siloed, and sometimes it's hard to know kind of how to query them or how to access them. And so I think this gives you like a good way to access them all at once. You can run different models. You can run a cell model. So this is a muscle model. And that muscle model gives you an output. So it actually gives you a graph. I can run a muscle model with parameter 4.1 PA, and it gives me this finished simulation. So it's actually, I don't know what the injection, this is injecting something into the muscle. There's a neuron model where you have an injection level as well. You can run the neuron model. So it's running their models and it's doing this. So it's giving you like electrical activity in SI units over time. All right, now let's go to some papers. And there are two papers here on what I'm calling developmental trajectories as inner product space. And this is where in these two papers, they basically treat developmental trajectories as sort of this inner product space. And I'll describe what that is in a minute. So when we talk about an inner product space, usually what we mean is like we have these two functions that intersect, and we have this angle at which they kind of form once they cross and go in other directions. So this is function A and function B. All right. And this might be a graph with time. So you have, and then some variable here. And so this is the inner product space here. And you're, what does this have to do with development? This looks like something that's mathematical. Well, the idea being that in development you have these trajectories. So if you think about like the epigenetic landscape of Waddington, you have this landscape that kind of comes down like this, and you have these what he calls this process canalization or developmental channels. Basically, these are developmental trajectories that sort of get reinforced by the environment, by genes interacting, genes being expressed, and the sort of the body plan of the organism and so forth. So you start up here at the top, which is like the single cell embryo, and you go down this path and you end up with these sort of switch points or decision points where you go down different channels and you make your way down to the bottom where you end up in a unique location sort of at the end of development. So this is adulthood, or I guess maturity. And this is the juvenile here, juvenile stage, or the embryonic. And so you can see that there are these unique developmental pathways that kind of result from this process. You have these branching points, and you have these developmental pathways. So these developmental trajectories are sort of, you can plot them out like this. You might have one that goes like this, one that goes like this, one that might go like this. And so you can see where you get these inner product spaces in different places. So you can use these to sort of understand the divergence of development. So if you have like something like this trajectory here versus this trajectory here that I've kind of defined in these arrows, those are different. Those are going to maybe not have an inner product space. They might diverge like this. They'll have an inner product space at the origin point, so kind of like right here, but not later on, as opposed to maybe a trajectory that intersects with this. So you might have a trajectory that kind of comes like this. If you have polyphenisms, oftentimes you'll have alternate paths for the same organism depending on its environmental context. So if we have a polyphenic organism, we might have something that maybe goes down this route during normal environmental times and this route during like stressful environmental times. And that gives us a inner product space here. So these are developmental trajectories. And then we have an inner product space. And this could be due, this is, we'll see in the papers that they use these for different reasons. It could be like morphological, it could be life history, it could be even gene expression. A lot of times gene expression patterns, people will use this to interpret like next-gen sequencing data and things like that, where they can form a trajectory or a vector of some type, and then two vectors have a crossing point, and then they have an inner product space, and you can determine that, you can define that, and you can quantify a lot of these changes in gene expression. So, but of course, those don't have a developmental context. What I'm saying is this is the broader developmental context of all of this. So let's start with this paper here in BioSystems. This is called The Indispensable Role of Time in Autonomous Development. This is by William Letsou. He's from New York Institute of Technology. And I actually reviewed this paper for publication. I thought it was good. It had, we had a conversation back and forth about what we were talking about here, and nailing down all the theoretical points. But I think it's a very good paper, especially for some of these issues with respect to quantifying development. So let's see if I can read the abstract. It's a little hard to read, but the abstract reads, advances in single cell analysis have led to a picture of development largely in agreement with Waddington's epigenetic landscape, in which a cell fate is determined by its basin of attraction in a high-dimensional gene expression space. So again, those landscapes I was showing you, you can think of those as sort of like dynamical systems. The channels or the canalization process, it goes down these sort of, you know, long, thin retractor basins where, it's easy to kind of find a path forward once you make a bifurcation, and then you end up at the bottom. So it's kind of like if you roll a ball down a hill and it follows a path of least resistance and you keep rolling balls down the hill, it tends to form a little groove into the ground, and it makes it easier to go down that way the next time. And then pretty soon your ball gets buffered into that canal or that channel, and it always goes down that route unless it's really forced out by some sort of energy. And so, same thing with development. We usually have these canalization as a process of buffering. It's phenotypic buffering, where the phenotype is sort of kept within the constraints of that particular path, and it requires maybe something like a macromutation or some other type of perturbation to drive it out of that trajectory. So that's kind of where we're going with thinking about Waddington's landscape and these basins of attraction and this sort of high dimensional gene space Waddington's landscape. Conceptual gaps remain as to how a single progenitor can simultaneously generate multiple endpoints and why time should be required of the process at all. We propose a theoretical model based on Hamiltonian mechanics of n-dimensional rotational motion, which resolves these paradoxes. We derive the result that systems which become different for themselves over time must initially move in a direction not towards their ultimate endpoints, and propose that this process of resolving ambiguity can be quantified in an information theoretic sense by the volume subtended in gene expression space, by the trajectories taken by the system toward those endpoints. So basically, he's describing the process I just diagrammed out. It was a little bit less verbose, and what I showed you was kind of like the mathematical interpretation and then the developmental context. We discussed the implication of this theory for the analysis of single-cell gene expression data in studies of development. So I've highlighted a couple of points here. The first one is that he talks about the landscape model, and he's kind of getting at this sort of maybe limitations of the landscape model and the sort of the benefits of the landscape model. And so he's been talking about the landscape model, and then he gets into this discussion here. So all right. So furthermore, the landscape model has even been refined to capture some aspects of indeterminacy and simultaneity. Wang and colleagues first introduced the so-called curl flux force to account for limit cycles and non-equilibrium, non-gradient behavior. So these are things that are non-gradient, non-equilibrium. So landscape models, if you see the connection with machine learning, we're dealing with a gradient basically. And things move down a gradient and down the landscape. As they move down to the bottom, they sort of minimize their developmental potential, or you start at the top, you can go in any direction you want, you can take any set of paths you want. But as you get farther and farther down the developmental landscape or the epigenetic landscape, your decisions have already been made, your potential is less because you've taken all the available paths. And you end up at the bottom where you have maturity and you have that phenotype that you're sort of limited to. You're buffered into that attractor basin, and it's very specific for your genetic and environmental context. So that's kind of what they're talking. They're actually talking here about some of these issues with respect to non-equilibrium, non-gradient behavior, These and other authors also rejected the idea that surfaces could be static. So this is where the surface is dynamic. It's always changing, depending on differences in the genome, depending on differences in the environment. By their demonstration of how tuning parameters in the coupled differential equations governing the mutual expression levels of various genes may generate bifurcations in the landscape. So again, the Waddington landscape, it's all underlined by gene expression, like coordinated gene expression, and there's a picture, which I think I've shown in a previous meeting, where Waddington's drawn the landscape and then he's drawn like a network of genes underneath. And when he wrote the book, they didn't really understand genetic regulatory networks. So it was interesting that he had this conception of it. But basically, it's controlled by genetic regulatory networks underneath the surface. And that's what determines these switching events, these bifurcations, and how these things are buffered into these non-equilibrium attractors. This is where sometimes we have progenitor states that become unstable. Sometimes, we get this buffering. There's a lot of sort of a role of stochastic fluctuation in gene expression. And this is something that's also captured in this model. And this is an interesting point here. So the elucidation of the role of stochastic fluctuations in gene expression play a bias in biasing a pair of dividing cells into alternate fates was another important experimental and theoretical advance, for it provided a non-teleological mechanism by which such bifurcations could arise. So this is interesting. If we go back to our landscape, we saw that there were these bifurcations. And you might ask, well, how do they happen? And I said, well, it's coordinated gene expression. It's the role of environment. But it still doesn't explain why these are sort of adaptive. I mean, why they should be adaptive. I guess if you generate a embryo that isn't functional, that kind of selects for things that are functional. But also that there's a sort of a temptation to say, well, it's a teleological process. That development unfolds by some sort of, blueprint or plan. And that's actually not what we know that not necessarily to be true. So how do we get these bifurcations in the landscape? How do we get to phenotypes that are alternate phenotypes, but also are functional phenotypes? And so, one way to do that is to, of course, have these stochastic fluctuations in gene expression, which allow for cells to bifurcate into alternate fates. It doesn't require any sort of architecture or blueprint. It just requires this sort of noise with, gene regulatory networks, with interactions with the environment, and so on. All right, so more recently, the introduction of RNA velocity as a method for tracking the instantaneous direction single cells take in a gene expression space has led to a new understanding of the Waddington landscape in terms of vector fields. So this is where we get into these inner product spaces. So we think about RNA velocity as sort of like what is the RNA complement over time, and how fast are things being expressed? And so we can actually measure all that as sort of this velocity vector. And so we want to know kind of what in gene expression space, which is abstracted from even from the epigenetic landscape, because it's just describing gene expression as a vector and then gene expression change over time as a vector. So we get this sort of velocity approach, which is very similar to like a velocity approach of motion, where you're looking at sort of the position and then the derivative position. We have vector fields, which are the velocity of RNA. And we have this where they have these inner vector spaces because they cross each other. And so, or they're different from each other. And you can measure that difference in that way. This is an interesting approach. So people have used vector fields before. Corson and Sigia 2017, Schmidt 2020, Q 2022, and Gao 2023. In these studies, there is shown a clear flux in different directions for progenitors to differentiated cell fates. There is even evidence that cancer cells metastasized by both their reversing and recapitulating the developmental trajectories taken by nearby cells of a related lineage. So this is where, you're kind of, they're either replicating the developmental trajectory or reversing the developmental trajectory. And these are cancer cells. So these are cancer cells that are growing much like a tissue with the types of cells that were hijacked by the cancer. Um, so they also have this sort of developmental trajectory. So this is interesting. And also the dedifferentiation proceeds along the reverse of the paths taken during differentiation. So sometimes cells dedifferentiate. You have a number of model systems where cells can go from, say, like a muscle cell or some type of neuron, and then reverse course and become like basically a stem cell. When we have cellular reprogramming, we kind of do this dedifferentiation process. It's a little bit different than what we see in nature, but you have these cases where cells will dedifferentiate and they'll just become like sort of multipotent again. Basically, they pop out of their differentiated state and back into a multipotent state. And so it's interesting that they follow the same sort of, they get buffered into the same sort of developmental trajectory. It's just that you're running it in reverse. Collectively, these results suggest that the tracks laid down in the landscape are well defined and stable, and more broadly, that a combination of deterministic and stochastic dynamics underlie cell decision making during development. So this is a combination of deterministic and stochastic dynamics. We have these deterministic dynamics in these canalization trajectories, and then we have the stochastic component that can pop them out of the trajectory. It can make these bifurcations happen. It can also reverse course and result in dedifferentiation or something going down the cancer pathway. So it's really interesting. So then, he talks a little bit about, Waddington's hypothesis of canalized development. And then, if correct, this analogy would so he talks about the hypothesis of RNA velocity vector fields, which are effects, not causes of changes in gene expression. So this isn't a cause of gene expression, it's an effect. So if I look at the velocity vector, it's basically what's being expressed over time, how fast it's being expressed. And so you're looking at the effects, not the causes. If correct, this analogy would resolve the paradox in the Waddington model, forced upon us by the existence and uniqueness theorem of dynamical systems theory, namely, the stipulation that a single progenitor cell have but a single fate allotted to it by the integral curve traced from its starting point, which are cells, which under the gradient model would not be permitted to start at the end, despite their knowing the end at the start. This would, under the new model, only learn their ultimate endpoint upon achieving it. Similar notions have been recently expressed that cells do not contain a blueprint or program in their DNA, but rather a generative autoencoder, which is set up to adapt and constrain the several potentialities of the cell without being specified ahead of time. This is Mitchell and Cheney, 2024. So this is what I was talking about, that, there's this idea in developmental biology that there's this sort of blueprint or program that tells the cell where to go to the endpoint from the beginning. And so the cell knows kind of what it needs to do, it just needs to get there. Now that, of course, invokes teleology. It invokes a lot of sort of assumptions about what's going on in the cell, what's going on in the DNA. But what he's saying is that there's some sort of generative autoencoder. And so this is the paper Mitchell and Cheney, which is here, and this is the genomic code. The genome instantiates a generative model of the organism. This is a archive preprint. This is Kevin Mitchell, the first author, and this is the genomic code. I think we went through this preprint before, but basically using the sort of generative autoencoder, variational autoencoder metaphor to describe what's going on in terms of gene expression and that combinatorial gene expression you see in Waddington's landscape. So again, this might suffer from the same problems as we have with the blueprint model. Namely, we're using a metaphor that's sort of current and even hip to describe what's going on in the cell when we don't maybe actually know what's going on in the cell. So to recap, in a variational autoencoder, you have this latent space. The latent space sort of describes all the potential changes, and then the cell would reference that latent space, which I guess would be the genome, and all of its states of instantiation, all of its possible states of instantiation, and then it would select a subset of those to go forward with to achieve what it needs to achieve. It's a lot more plausible, I suppose, than the blueprint model, which basically implies that everything is sort of preordained. Okay. So the next paper is this PNAS paper, utilizing developmental dynamics for evolutionary prediction and control. So this relates to what we were talking about in the first paper. Can we predict evolution? Can we control evolution? Well, can we use the developmental dynamics to do this? So with this journal, we have a significant statement and an abstract. I'll read the significant statement first. So variation in traits is the raw material for evolution. Yet, the complexity of development makes it daunting to predict, control, and engineer evolution by manipulating the generation of phenotypic variation. So that's what we mean by predicting and controlling an evolution, or engineering evolution. We want to manipulate the generation of phenotypic variation. Much like what we want to do with the epigenetic landscape, we want to start with a single cell and move down to a very refined phenotype. By representing development as a dynamical system, we build a bottom-up theoretical framework to predict the effects of genetic and environmental perturbations on the resulting traits. We find conditions for when different perturbations result in concordant changes in the traits and show how this could be used for evolutionary prediction and to steer evolution in a preferred direction. So that's the significance. And then the abstract says, understanding, predicting, and controlling the phenotypic consequences of genetic and environmental change is essential to many areas of fundamental and applied biology. So we want to be able to do this understanding, predicting, and controlling aspect of this. We don't want to just kind of watch it happen. We don't want to have this sort of process that's, an opaque process. We want to be able to manipulate it. In evolutionary biology, the generative process of development is a major source of organismal evolvability that constrains or facilitates adaptive change by shaping the distribution of phenotypic variation that selection can act upon. So if we think about our, landscape again, we have these trajectories. That is sort of like, maybe like stripes on a distribution. And we want to be able to kind of shape that distribution in ways that is, understandable. Whereas sometimes if we're doing genetic engineering, we want to shape it in ways that's functional. And of course, when we do artificial selection, like domestication or something like that, we likewise get that sort of effect. While the complex interactions between genetic and environmental factors during development may appear to make it impossible to infer the consequences of perturbations, the persistent observation is that many perturbations result in similar phenotypes. And this indicates that there is a logic to what variation is generated. So that kind of almost speaks to the model of, the variational autoencoder. That it's, there's this variation that's sort of latent, but then there's some structure to how it's, put into practice in the world. Here we show that a general representation of development as a dynamical system can reveal this logic. We build a framework that allows predicting the phenotypic effects of perturbations and conditions for when the effects of perturbations of different origins are concordant. We find that this concordance is explained by two genetic features of development, namely the dynamical dependence of the phenotype on itself, and the fact that all perturbations must affect the developmental process to have an effect on the phenotype. So this is interesting because, we have, of course, in development, the phenotype, starts off at a single cell, and we talked about how you get these bifurcations. And you increasingly, as you go down the slope of the epigenetic landscape, you become more locked into certain phenotypes, simply because you've already made those decisions. And you can de-differentiate your cells, you can do that, but you have this sort of increasingly constrained landscape. So phenotypes are dependent upon their sort of prior developmental history. And then the fact that all perturbations must affect the developmental process to have an effect on the phenotype. So if it doesn't affect the process itself, it doesn't really show up in the phenotype. We apply our theoretical framework to classical models of development and show that it can be used to predict the evolutionary response to selection using information plasticity and accelerate evolution in a desired direction. The framework we introduced provides a way to quantitatively interchange perturbations, opening an avenue of perturbation design to control the generation of variation. So this is interesting. They kind of talk about this process that generates variation. It's known as development. Notoriously complex and nonlinear interactions between genes, cells, tissues, and environmental factors during development make it difficult to grasp the phenotypic consequences of genetic and environmental perturbations. Indeed, the diversity and complexity of developmental systems could be feasible, or could be taken as evidence that a priori influence on the consequences of perturbations rarely will be feasible. So, we look at the diversity and complexity of developmental systems, and we can't necessarily say that there's any sort of a priori influence or blueprint or teleological forcing or any of that. A pessimistic conclusion is therefore that the best one could hope for is to demonstrate that generative processes in principle could impact evolutionary trajectories, while studies that demonstrate how development affects evolution remain a collection of idiosyncratic case studies. This perception that generative processes are intrinsically unpredictable and that selection is the only reliable force in evolution is also reflected in biotechnology and medicine, where attempts to direct evolutionary processes emphasize control over selective regimes rather than control over generative processes. So this is interesting. We're going in the same direction in terms of generative processes and how they influence phenotype. So their methodology here is interesting, but essentially what happens is they end up with these different developmental trajectories. And then, of course, by virtue of having these developmental trajectories that diverge, that intersect, that have features of sort of this generative process, you get this sort of these inner product spaces, and this is the same sort of approach, or a little bit different approach than we saw in the last paper, but in the same direction, I think. So this is figure 1 from this paper. This is a general framework to study the phenotypic effects of perturbations of different origins. So the top image here, it looks like 2 developmental trajectories that are diverging, and you can take a time slice through that and look at these other sort of graphical representations down here. So the one at the top here are the developmental trajectories that go over time. And then you have this time slice here where these trajectories cross through the time slice. And then you can see that these trajectories, the black and the purple one respectively, are shown on this graph with respect to the perturbations. So on top, which is this large figure with the trajectories, the reference developmental trajectory, which is at the bottom, which is the black one, in black, and the perturbed trajectory in purple through developmental time t. So the purple one is the perturbed one, black one is the reference trajectory. The panel in the middle shows that at any given time, and this is the panel in the middle here, the effect of the perturbation on the trajectory given by the sensitivity vector, which is this sensitivity vector down here, or actually this purple one, I believe, is a linear combination of the columns of the matrix A to the power of -1. That's the transpose, or A of -1 of the reference. So the three panels at the bottom show the sensitivity vectors for different perturbations at a given developmental time, and that these are linear combinations of the columns of the same matrix. So S1 and S2, which are these two, are largely aligned, but S3 is not. So you can see if we zoom in, these two are largely aligned, and this one is not. So you can see that you have this sort of intersection of these different trajectories showing the forcing mechanism and showing how they sort of operate at that time slice. Okay, now they talk about actually explicitly about these inner product spaces and how they're doing this in the context of aligning the effects of perturbations of different origins. So we saw in the last image up here where we have these intersections of the reference and the perturbed developmental trajectory. Now they're going to talk about sort of the formalism and how we might understand this in terms of these inner product spaces. So we now use the formalism introduced above to study the relationship between the phenotypic effects of perturbations of different origins. Given two developmental parameters, we say that their effects are totally aligned if the associated sensitivity vectors have an angle of 0, meaning that the two perturbations result in phenotypic changes in exactly the same direction. So we saw that there were different angles of sensitivity where the two perturbations will go in the same direction. More generally, we say that there is some degree of alignment if the two sensitivity vectors have an angle that is significantly smaller at a given confidence level than the distribution of angles between independent random vectors of the same dimension, which approaches 90 degrees as the number of dimensions increases. So basically, if they're totally aligned, they're an angle of 0. We can say that there's some degree of alignment if they have some angle of, they're subtended at some angle, but they're not, maybe they're under 45 degrees. And then the distribution of angles between independent random vectors. So these are just random vectors that have no developmental trajectory. If you perturb random vectors, they kind of do whatever they want and they don't really have, any sort of alignment at all, which approaches 90 degrees as the number of dimensions increases. So this is where we have high dimensional spaces where we have random vectors versus high dimensional spaces where we have developmental trajectories. So they talk about figure 1, giving an example for 3 developmental parameters, which can correspond, for example, to the affinity of a cofactor modulating gene expression, temperature and salinity, or other types of parameters that are not genetic or environmental. So we can map these perturbations or we can map these developmental parameters to different sorts of things going on in the world. So, this isn't just like an exercise in mathematics, it's an exercise in like saying, what are the effects of this thing? The effects of modifying each of those parameters at time t is given by the vectors for 1, 2, and 3, and the angles between them determine alignment. From equation 5, we know that all the sensitivity vectors can be written as linear combinations of the columns of the same matrix, the inverse of the Jacobian, where each column is weighted by the elements. This provides the basis to derive sufficient conditions for alignment between the effects of different perturbations. So if two perturbations have a dominant component in the direction of the same column of the inverse of the Jacobian, then the perturbations will be aligned. That is, there will be some degree of alignment between perturbing 1 and 2 if there is I, such that this expression Furthermore, there will be total alignment between the perturbations if B and B1 and B2 are proportional. So we have this sort of, this mathematical model. And, we can extend this to reaction diffusion models. So reaction diffusion models are a set of models of pattern formation that have been widely used to represent diverse developmental processes, including digit formation and hair follicle placement. These models consist of a physical representation of the tissue and a set of molecules called morphogens. Morphogens diffuse and interact with the tissue, leading to the emergence of patterns as they accumulate in specific spatial regions. So they will use the Gray-Scott model, which is a reaction diffusion model, to illustrate how we can study the alignment of phenotypic effects for different origins using the framework introduced in the previous sections. So this kind of goes over the developmental function for the Gray-Scott model. X1 and X2 are these dynamical systems that describe the cellular concentrations of morphogen 1 and 2. And then the components D1 and D2 within the equations X1 and X2 respectively are their diffusion rates to neighboring cells, which is described as this gradient. The gradient is the Laplacian operator, F is the production rate of morphogen 1, and K is the rate of degradation of morphogen 2. So we have morphogen 1 is being produced, degradation of morphogen 2. These parameters have been shown to have genetic basis in some systems, so the perturbations in these parameters can be understood as resulting from different genetic changes. So this is where we have genetic mutations or something where we're changing the concentration of morphogens and how they're diffused. So they model this on a grid of 50 by 50 grid of cells. And each of these cells is a given amount of 2 morphogens. So we can do that to sort of approximate the genetic variation of the cells. So they run this simulation. They run the simulation with small perturbations in the developmental parameters and compare the resulting phenotypes. So we'll see in figure 2 what that looks like. The phenotypic effects either show a decrease in K or an increase in F. And this is up here, the developmental parameters. These are largely they should largely aligned, resulting in the formation of a set of connected dots rather than dots in the reference unperturbed phenotype. So we'll see what that looks like. So the perturbed phenotype should not be identical because the angle between SK and SF is not 0. So now they want to build a genetic regulatory network to study the alignment between phenotypic effects of perturbation. We use a common representation of gene regulatory networks found in the literature with continuous representation of time. And the conventional formulation of these models, the phenotypes are the expression levels of end transcription factors that regulate each other's expression. We represent these phenotypes at states X, so they're NX phenotypes, with Xi being the expression level of the ith gene. This function f giving the change in states during development for this example is encapsulated in this equation, where we're looking at the ith jth element of the matrix and gives the regulatory effect of gene J and the expression of gene I. So gene J is regulating gene I. We have activation, inhibition, and no interaction. The expression of each gene can also be activated or inhibited by environmental inputs U, and so we have U of N. Gene expression follows the Michaelis-Menten dynamics of the coefficients Ki, and gene product has a degradation rate given by mu I. In this way, the developmental parameters for this example are here. So this is basically what they're doing with the gene regulatory network. So this is figure 2. This is alignment in a reaction diffusion model. So on the left, we see in purple the angle between 30 degrees and 90 degrees between SF and SK. Remember we said SF and SK don't equal 0. They're not totally aligned, but they're going to vary between 30 and 90 basically. This is showing this through developmental time, so you have a lot of fluctuation early in developmental time in terms of this angle, and then it settles down after, I don't know, 300 units of developmental time. So this is, again, the angle between SK and SF, those vectors, and then we have this random, which is a control, which is at 90, just about 90 degrees throughout developmental time. So it's like unalignment and then what's aligned, relatively speaking. So you can see that the angle kind of is quite high early in development, reaching 90 at some point, and then it settles down to an equilibrium at about 50 degrees or so. Yeah, so after a transitory period with oscillations following the introduction of the perturbation, so that's what this is, where the perturbations are being introduced early. The angle between SF and SK is significantly smaller than the angle between random vectors, which is this orange line, orange concentration of points at the top, indicating alignment between the phenotypic effects of perturbing K and F. The three panels on the right, which are these three here, show the phenotypes plotted as the concentration of morphogen 1, with higher concentration in lighter color for the reference developmental parameters, perturbed K and F respectively at developmental time 500. So they're looking at the end of this graph here, at the end where it's settled down, and they're looking at the reference, which is this, I guess, where the dots are aligned. You can see that. You have perturbed K and perturbed F. And perturbed K being in the middle, perturbed F at the right. As indicated by the small angle between F and K, the phenotypic effect of the perturbations is smaller, resulting in connected dots, as opposed to the dotted pattern of the reference. So the reference shows this complete sort of Gray-Scott morphogenesis, where you have the independent dots aligned. The perturbed K, you see the, sort of that the dots are connected through this sort of drift. So sometimes you see some connections, sometimes you don't. I mean, this one here doesn't look connected. This one here doesn't look connected, but you get this connection. And then in perturbed F, you get more connection. It's just kind of a different, it looks kind of blurry in K, more blurry than K than F. But basically, you get these connected dots as opposed to the dots in the random pattern. So basically, the reference has no perturbation. K and F have a perturbation. That perturbation results in drift of the morphogen between the dots, and it ends up with this. So you basically get the connected dots, which are sort of this regular pattern, and then you get these sort of almost like stripes, which are a little bit different. So, I don't necessarily know how to interpret that other than to say it's an interesting result. So finally, let's talk about figure 4. So figure 4 shows these trajectories. It shows fitness over a number of generations. So this is evolutionary. And this is showing how plasticity predicts evolvability and how this can be used for evolutionary control. So you can see that you have these trajectories here. You have these trajectories in evolution. So let's look at this. Panel A, which is on the left, shows an example population of the left, right, and up-down sets in orange and black respectively. Dot represents unperturbed individuals, no environmental inputs. So you can see the dots underlying that. And arrows represent the direction of the change in steady states when an environmental perturbation is introduced in one of the first two states. Panel B in the middle shows the evolution for left, right, and up-down populations towards an optimum located at x3, x4, 12.5 and 7.5, represented with the purple arrow. So that's up here. The left-right populations in orange outcompete the up-down. Transparent lines are the average among the 25 evolutionary lines. So you can see these transparent lines initiated from a single individual for each of the 15 populations in each set. Panel C on the right shows that an additional mutational input directly on the first two rows significantly accelerates evolution towards the optimum of the left-right populations. So the fitness is increasing faster when you have an additional mutational input. So these are numeric simulations. They're using this model to sort of, look at developmental perturbations, look at how they're sort of affecting the developmental trajectory. Then they're looking at this in terms of evolution of development, looking at single perturbations, and then looking at multiple perturbations. Okay, so I think that's a pretty complicated paper, and it's something that I will link to in the description of the video. But I think that gives you an idea of how people are using these sort of mathematical tools to look at development and some of the problems of development. So that's all for today. Thank you for paying attention, and I hope you learned something.